everyone, my name is Lisa Markovchick and I am a PhD candidate at NAU in Flagstaff. And I am here to talk to you today about habitat and wildlife and what does mycorrhizal fungi have to do with that. So you might wonder why, when you guys probably care a lot about these guys, I want to talk to you about these guys. And I'm going to use the southwestern willow flycatcher as an example today. So we know that swiffle fecundity is declining due to nest heating. And we know that tamarisk and/or mixed vegetation is hotter and drier than native vegetation. And also that we're staring down the barrel of a lot more warm and dry weather due to climate change. And interestingly, mycorrhizal fungi actually contribute to water and evaporative services in the local area that they're found. So on the top left here, you can see an illustration just of the stomatal opening and a plant leaf. And we're just trying to illustrate the fact that mycorrhizal fungi actually help plants moderate their water use efficiency through a few different means, but particularly through modulating that stomatal opening and closing. And then on the bottom right, you can see in the little call-out box number 2 there are examples of mycorrhizal fungi transferring water. So on the left, transferring it within the mycorrhizal network. And then on the right-hand side of that circle, leaking it out into the upper soil layers, as is pictured below. And what you see below that is just a hyphal tip, where the water has been stained with a fluorescent stain. So you can actually see the hyphal tip in fluorescent green, as well as a little drop of water coming out of it. So before we go too far, let's just talk about what Mycorrhizae are. They're an ancient fungal symbiosis with nearly all land plants. They actually helped land plants move to land. And there's 2 main types that are relevant in our riparian communities, particularly for cottonwoods and willows, because they use actually both types, but also for any of the other trees in the riparian community. And in many of our habitats, they'll usually use either one or the other. So AMF is our buscular mycorrhizal fungi, pictured on the bottom left there inside a plant root cell. And then EMF or ectomycorrhizal fungi, which occur on the outside of the roots. And you've seen some examples of those already, and there's one right there. These guys all receive photosynthate in exchange for a variety of resources or services that they provide to the plants. And that list of services includes things like boosting survival, growth, resiliency in the face of stressors. Again, the water services that we just talked about, so providing water access and helping improve water use efficiency for the plant, providing access to nutrients and improved plant health, also providing a variety of techniques of pest control, and helping to reduce the toxics that are absorbed from the soil to the plants. I could go on, but let's stop there. The important piece is that these impacts on all of these services from mycorrhizal fungi are not negligible. They are very frequently 50% or more impact on, for instance, rates of survival or growth and so on. However, as we've begun to get more familiar with the kingdom fungi, which is still relatively underexplored compared to other kingdoms, we have learned that land use changes and different management actions affect mycorrhizal communities too, both in diversity as well as abundance. So for instance, invasive vegetation, and I'm going to talk particularly about tamarisk, but also things like habitat fragmentation, land use change, fuel management activities, herbicide application, many of the things that we do every day and often have other types of beneficial responses and that's the reason we're doing them, can impact our mycorrhizal communities. So you may have seen some of the results from my field trials. I'm just going to talk about it briefly. We worked out on the Little Colorado River doing tamarisk removal and cottonwood and willow restoration. And we set it up in this way. So we had either local cottonwoods from a nearby population or the site itself. And then we had what we called transfer cottonwoods, which were from a slightly warmer area, slightly further away from the site that we were restoring. We had areas that either had a tamarisk history and soil legacy or did not within the site. And then we either added a sort of live soil inoculum of mycorrhizal fungi from a site that hadn't had tamarisk and was more intact. And we either added that or didn't add that to those treatments in a fully factorial design. And what we found was pretty striking. So tamarisk legacy definitely reduced cottonwood survival, and the mycorrhizal inoculation improved survival, but the treatments and the tree source interacted. So I wondered, what values and services are we losing for plants, habitats, and other dependent species when mycorrhizal fungi in the community decrease? Can we restore those values as well by restoring the mycorrhizal community? So during the pandemic, we have been busy with a greenhouse trial, and it was basically set up in the exact same way, using the same tree genotypes and the same soil, but just in the greenhouse where we could control a variety of factors a little better, such as soil moisture and things like that, and make sure they were consistent. Borrowed a gas exchange system, the Leghorst 6800, so that we could take some gas exchange measurements and look at the ecophysiology of the plants. And what we found was that for local trees, tamarisk legacy reduced the water use efficiency. So on the left-hand side of the graph, you can see the treatments where we did not have tamarisk soil. So sort of the status quo, if you will, both inoculated or not. And on the right-hand side of every graph, you're going to see the plants that were planted in the tamarisk legacy soil. Those dark bars are always going to represent mycorrhizal inoculation. The light bars are without mycorrhizal inoculation. And what you see here is that in tamarisk legacy soil, that light bar of water use efficiency went down. And then when we added mycorrhizal fungi, that water use efficiency went back up. Survival showed a very similar pattern. Why might that be? So tamarisk legacy seemed to actually cause the trees to produce beyond their means. So if we look at a graph of the photosynthesis, we can see that on the right-hand side of the graph, that photosynthesis in the tamarisk soil shoots way up without mycorrhizae additions, and then sort of goes back to the normal level when we add that mycorrhizal inoculation. For the transfer trees, the results were actually almost exactly the opposite. So again, on the left-hand side of the graph, sort of the status quo water use efficiency. On the right-hand side of the graph, we can see what happens in the tamarisk legacy soil without mycorrhizal inoculation. That water use efficiency shoots way up. When we add mycorrhizae, it brings it back to that status quo level again. And we can see that the survival is also tracking that pattern. This seems to be because tamarisk legacy is actually shutting down the plant production without mycorrhizal inoculation. So you can see here in the tamarisk legacy soil, that photosynthesis, the light bar on the right-hand side, goes way down. When we add mycorrhizal inoculation, it bumps back up to sort of that status quo level again. So in summary, due to the tamarisk legacy and reduced mycorrhizal communities, local trees overproduced, reducing their water use efficiency and having negative impacts on their survival. Transfer trees did the opposite. They actually shut down in that tamarisk soil, which improved their water use efficiency and survival, but would essentially shut down their photosynthesis and resulting growth. In both cases, the mycorrhizae actually stabilized the situation. So they stabilized both the photosynthesis and the water use efficiency while maintaining survival at a better rate. So why would trees react oppositely to this mycorrhizal loss and or restoration? What we've been learning from the literature is that the pairings between the trees, the soil, and the fungi really matter. And so in this case, transfers were better paired with the mycorrhizae, and locals were better matched with the soil. Also, the plant growth strategies probably differed because the ones from warmer sites would have had a slightly different growth strategy. Appropriate mycorrhizal inoculation basically really matters. And when I say appropriate, what I'm talking about is this pairing between plants, soil, and mycorrhizae. So we can actually get neutral to negative effects with poor pairings. And we just really need to think about those hard and long when we do our mycorrhizal inoculations and restorations. So I wanted to know if tamarisk reduced mycorrhizal communities cause plants to shut down production or die, then shouldn't mycorrhizae boost swiffle habitat suitability, and would we be able to see that in a habitat suitability model? And so James Tracy, who's presented his swiffle habitat suitability model at this conference in previous years, it's a GIS-based site-specific model, went ahead and entertained my questions and added a restoration project of cottonwoods and willows to an area in the model that was previously tamarisk. And what we did was we went ahead and added the survival parameters and growth parameters from the literature for cottonwoods and willows. And then we also added on the bottom here, you can see some of the references for what mycorrhizal inoculation did for plants, whether it was biomass or survival and those kinds of things, to the model. So we incorporated that into the model, and we had a treatment without mycorrhizal inoculation and with. And you can see on the left-hand side here that with mycorrhizal inoculation, after 6 years, we actually have 1.2 hectares of suitable habitat at this site for swiffle, and that would be corresponding to that purple line on the graph where you can see the canopy cover increased much more quickly than the blue line, which is if we did not have mycorrhizal inoculation. And in fact, after 6 years post-restoration, if we did not include mycorrhizal fungi, we still had 0 hectares of suitable swiffle habitat. So invasive reduced mycorrhizal communities also reduce plant survival and growth, and as a result are going to be reducing suitable habitat for other species. And we're just talking here right now. We haven't added anything but growth and survival metrics to the model. So we're purely talking about growth and survival. We're not even yet including things like what effects that has on shading, what effects that has on canopy cooling with the evaporative services being provided. Also, appropriate mycorrhizal restoration is stabilizing plant survival and growth and increasing suitable habitat for other species. Again, just purely based on mycorrhizal impacts to plant survival and growth, not even yet incorporating shading, canopy cooling, evaporative services, and so on. And so just like these guys who all have very specific needs and requirements and perform different roles within the ecosystem, we need to remember that these mycorrhizal communities, all the different species and taxa in there, have similarly very different needs, roles, and provide different services. So as part of that, we're actually going to be using stable isotopes next with tracking water movement through mycorrhizal hyphae in an attempt to go ahead and identify which mycorrhizae seem to be accessing water and providing it to plants, and also to see whether the tamarisk legacy is actually reducing not just the mycorrhizal community and abundance, but actually negatively impacting the mycorrhizae that might be providing these water services. I just want to say a huge thank you to everyone for attending today. It's been a pleasure talking, well, to or with you. And this research would simply not be possible without an army of folks. I can't even get the list on the slide anymore. So I'm just very thankful to all of them as well. And also a note that if you're interested in incorporating mycorrhizal restoration into a project, I would love to hear from you. I'm trying to get us set up so that we can provide that service more regularly to folks, and I would love to chat with you about it. And again, thank you so much, and hopefully we have some time for questions.